all. Today's lesson is a 1-hour lesson on GCSE Biology, focusing on the infection and response recap. If you've joined the online school before, then this should follow on nicely from other lessons that we've been having in biology recently. If you haven't, then welcome. It's your first time and it's a good time to join because we're recapping quite an important topic. It'll give you some ideas of what you need to revise or go over. My name's Charlotte. I'm the biology tutor for the online school. I have been tutoring with my tutor for a couple of years now. I studied conservation and biodiversity at the University of Exeter, and I've completed over 150 lessons with my tutor. Okay, we also have Bola from my tutor on hand to give us any help with any technical difficulties or anything that we come across in the lessons, so feel free to ask questions per on the chat as well. If you do have any questions, the way I like to run my sessions is that if you look at the bottoms of your screens, you've got a Q&A button and you've also got a chat button. Both of them have little speech bubbles on them. If you use the chat button for answers to mini quizzes and things like that, and if you use the Q&A button for any questions that you've got about the topic, and what I'll do is I'll make sure that I've got that up and I can see any questions that you have that are coming in. Now, okay, so let's talk about today's lesson. So today's lesson is a recap of the infection and response topic. We've focused mainly on the AQA specification up until now, but we've made the content as relevant as we can to other specifications. So you will learn things that you need to know if you're not studying the AQA specification for GCSE. Infection and response has involved us talking about communicable diseases and pathogens, bacterial pathogens, viral pathogens, fungal pathogens, and protist pathogens and the diseases that they cause. We've also talked about the human defense systems. We've talked about vaccination. We've talked about antibiotics and painkillers. The discovery and development of drugs for treating those diseases. And then we've also covered monoclonal antibodies, plant disease detection and identification, and plant defense responses. So it's quite a big topic. We've covered lots of stuff. And what I wanted to do today was kind of see how much you've learned so far, have a look at any questions that you might have, and try and answer those. And we'll go through some past paper questions as well and do some exam technique. So the first thing I thought we'd do is a bit of an experiment, but I thought it might work quite well. So I wanted us to make kind of a mind map of what we think of when we think about infection and response. So what I wanted to do, activate this and then send you the link. So what I'll do is I'll copy the link and then I'll pop it in the chat. There we go. So if you go to the chat now, we'll see if this works. If you copy the link that I've pasted in there and put it into your browsers, you should be able to see a question. And that question should come up any second on the screen. So I've said, what are the key words that you think of for the infection and response topic? Can't see anything in your chat box. Oh, that's because, hold on 2 seconds. There we go, you should now. Okay, so if you paste that link into your browser and then answer the questions in there, you can answer as many times as you like, but just put in what the key words that you think of for the infection and response topic. And what it'll do is when you start putting those in, it'll start building up a word map on this screen. We'll see if it works. Good. Okay, pathogen. Excellent. Any more? Put in as many as you can think of. So what were the topics that we talked about? We talked about different diseases, different pathogens from different organisms. Good, nice. Virus is in there. That's one of the types of organisms that we talked about. Life, antibiotics, vaccines, really good. Antigens, brilliant. Antibodies, yeah, really good. Good to know the difference between those. Sometimes that's a bit difficult. Protist, good. What disease is caused by a protist? Loads of stuff coming in there. White blood cells, I'm assuming that's comes through. Tobacco mosaic virus is in there, really good. Lymphocytes, cells, vectors, yes, exactly. So mosquitoes are a vector that carry, what disease do they carry? Malaria. Phagocyte is in there as well. This is really good. Got bacteria in the middle there. Everyone's thinking of bacteria when they think of disease, which is interesting actually, because if you think at the moment, the disease that why we're all social distancing and why you're attending an online school is caused by a virus. And yet we still consider bacteria mostly when we think about disease. Okay, this is great. Any more in their symptoms? B and T cells, good. Infectious, yes. Host disease. Killer T and B cells, good. It's a really good understanding of the immune system in there. That's brilliant. Vaccines, phagocytosis, vectors are coming up lots now. That overtaken bacteria in the middle there. STD, yes, sexually transmitted diseases. We talked about those. Talked about the blood as well. This is great. This has worked really well. So what I thought we'd do is we'll answer some questions and things, and then we'll do the same thing again at the end and see if it's changed at all. That would be a good thing to do. Okay, so I'm going to stop this one now and head back to our lesson. I'll just close this one down. Good, so we had loads and loads of answers there. That's really, really good. Okay, so thinking about those topics there, what would be really good, and what I've kind of prepared first, is any questions that you have. So as a reminder, the first tutorial that we went through were communicable diseases. So that's where we talked about pathogens, viruses, protists and fungi and the different diseases that they cause. And then we went through, let's go back to this because it's easy to see, isn't it? Then we went through human defense systems. So we talked about our skin, we talked about enzymes that we have in our saliva, stomach acid, nose hairs, all of those things. We also talked about vaccination as kind of the next one on. Oh, and we talked about the immune system in between there, didn't we? White blood cells. Vaccination, how that works, what is vaccinated, and how to answer questions about vaccination. Antibiotics and painkillers. Drugs, their development and discovery, all the different testing that goes on, lots and lots of questions about those because it relates directly to scientific technique. And then monoclonal antibodies, plant diseases, and plant defenses as well. So those of you who were in those lessons, did you come away with any questions? I did mention at the end of our last lesson on plants we'd do this today. So did you happen to bring any questions that you'd like answered in today's session? And I've got all of my slides there so I can pull them in as needed. So if you popped your question and answer box, and then pop in any questions that you have about this topic. We'll have a go at answering them and then we'll move on to some past paper questions, I think. Oh, detailed one to start with. See if there's any more coming in. So the question was, oh, what happens if I press answer now? So the first question was, in a rough estimate, a percentage, how likely do you think it is that a vaccine will work in defending ourselves? So I'm assuming that you're meaning coronavirus specifically at this point, rather than the content. So it's really difficult to answer because although I'm a scientist, I'm an ecologist. So this isn't kind of my current area of expertise. But if we do get a vaccine, the thing that we need to work towards is herd immunity, which we talked about. So I'll pull up that slide. So we had this really good slide on herd immunity that I will pull up now. So herd immunity is something that we always talk about when we talk about vaccinations. So the two things that exam questions tend to focus on are what is a vaccine, the weakened form of the disease that we inject into our bodies to stimulate our immune system. And the way that that works in a population is by causing herd immunity. So we've got these three scenarios here that kind of explain to you how herd immunity is built up within a population. So in our first scenario at the top, mass infection. We've got lots of these blue people who are not vaccinated but still healthy. So that's like most of us, in fact all of us at the moment from coronavirus. And we've got a couple of people here who are sick and contagious, like we have now with the coronavirus. No one is vaccinated, and so the contagious disease spreads through the population. You see we've got this mixture of blue and red people again. We've got many more red ones who are not vaccinated, they're sick and they're contagious, and they're slowly infecting all of the not vaccinated but healthy people. So in that way, a disease spreads through a population, and the only thing that we can really do is take preventative measures such as social distancing, such as self-isolation, and try and prevent ourselves coming in contact with those who are sick and contagious. Then we develop a vaccine, which is what is happening at the moment. Scientists are working on a vaccine for the coronavirus quite quickly. Why do we need a vaccine and not antibiotics? That's a good one. Anyone wants to answer in the chat? That'd be a really good one to answer. Why do we need a vaccine for coronavirus rather than antibiotics? Anyone? Good. Antibiotics work at killing bacteria. Antibiotics cure, vaccine prevents. Good, but antibiotics don't work on a virus. Brilliant. Great, thanks guys. So that's exactly right. We need a vaccine for coronavirus because antibiotics won't work on a virus. Viruses work by sheltering in our host cells. And that means that any antibiotics or anything that we put in ourselves to try and treat it tend to attack our own cells, which is why we need a vaccine. We need to get to it before it gets to our cells. So if we've got a vaccine, we've got a few of these green people in the scenario now. So green people are vaccinated and healthy. And you see they're spreading among the vaccinated and healthy, the unvaccinated and healthy, and the unvaccinated and sick people. Some of the population gets vaccinated. Contagious disease spreads through some of the population. But you can see, so the red people are the people that have been infected, and you can see that there's less of them in this scenario than in the one above. So the vaccinated people here are starting to provide a bit of a shield to the unvaccinated but healthy people. So you see this green person stood between a blue and a red. So it's providing a bit of a shield so that the unvaccinated and healthy person isn't exposed to the unvaccinated and sick person. If the majority of people are vaccinated, it's a different story altogether. So we've got a couple of people who are sick and contagious. They are fairly well surrounded by vaccinated people. And therefore, it's very unlikely that the people who aren't vaccinated but are still healthy will come into contact with those people who aren't vaccinated and who are sick. Why would they not be vaccinated if they could be? Why would we not take a vaccination? So there are some people that have underlying health conditions that mean that they can't be vaccinated against a disease. And so that means that they rely on the other people who can be vaccinated to provide a shield to them for anyone who has caught the disease. So that's how herd immunity works. It basically means that there's enough people in the community that are, or in the population, that are immune to a disease, so have been vaccinated, to provide a shield for those who can't be vaccinated. It also contains a disease, so this red person here, it's not going to, well, this red person is more of a better example, it's not going to go any further than him. He's got vaccinated people all around him. So once the disease has run its course in the body and has either been killed by the immune system or has succeeded, then that source of the disease is gone and won't have passed any further. So that's how herd immunity works. It's been very, very successful for lots of diseases. We're all vaccinated for measles, mumps, and rubella when we're young, most of us, unless there's a special reason why not. And those three diseases are things that we often catch as children, and we're protected from those now. The incidence of those is very, very low in this country. It's also worked for things like tuberculosis. We don't have tuberculosis anymore, purely because of vaccination. Okay. Why can't we prevent people from catching the cold? That's a really, really good question. And it kind of links on to my next point, which links back to Summer's first question about how likely it is to succeed. So we don't vaccinate against a cold, mostly because we don't need to. We all get colds. It very rarely affects us badly. It's when we get flu that that can affect us more badly, and can kill people if you're old and vulnerable, or if you have a weakened immune system. But colds, in general, aren't too bad. It's good for us to exercise our immune system occasionally. So getting a cold and then getting over it is no bad thing. But colds are viruses that morph very quickly, that mutate, and so change their structure quite quickly. And that means that when a new cold virus comes into our system, our immune system can't recognize it. So those antigens that are on the virus aren't recognized, and therefore don't have any complementary antibodies in the memory of our immune system. So that means that we don't have any immunity to it. Now, it's a cold, so it's okay, we generally get over it. And our body generates that immune response a bit slower than it would have done if we'd have recognized it in the first place, but it gets there eventually, and then we move on. And that's okay. But no, we haven't been able to vaccinate against the cold because they are just changing all the time. And it's similar with the flu vaccine in a way. So the flu vaccine is something that you can have in the UK if you are over a certain age or if you have a weakened immune system. And the vaccine vaccinates against the strain of flu that the scientists have predicted is most likely to be caught. There are lots and lots of strains of the flu virus, but scientists have become quite good at predicting who... Sorry, that's my phone now. They've become quite good at predicting which sort will be most, which sort of flu virus is most likely to be caught that year, and so people are vaccinated against that. In the same way, COVID-19, if it's If it changes its structure after we developed a virus, then you may then catch it again. So that links to the next question, how likely it is that we can be vaccinated yet still catch a disease. If it's a slightly different form of the disease, then you might catch it. Hopefully, there'd be enough of immune response for it to be slightly quicker, but it might still be slightly unfamiliar. Someone else saying, COVID-19 is contagious. Can the vaccination effects be contagious? So when you have a vaccination, sometimes you do get a little bit of a response, but what you're vaccinated with is a weakened form of the disease. So it's not the full form of the disease. So if we have a look here, this was the slide that we talked about. So you have an altered form of the pathogen put into you. Your white blood cells then release the complementary antibodies to the specific antigen proteins that are on the surface of that weakened form of the disease, the pathogen. And then they clump the pathogen together, and the phagocytes can come along and engulf and destroy it. So you might have a little immune response, so you might have some sort of symptoms, but they'd be very, very light. You might just be a bit tired. Your body's working a bit harder than normal, but they wouldn't be contagious. Why are there anti-vaxxers? That's another really good question. So people who are against vaccinating have lots and lots of different reasons for their choice. Sometimes people are against vaccines because they've read that they might be linked to autism. There was a study that linked vaccines to autism, but it was found that the scientist who did it was cheating. He'd fabricated the results and they weren't true. And it had got past lots and lots of other scientists on the way. He'd done a good job of cheating, but he had. So that paper was then pulled down. Those results don't stand. So vaccines haven't been linked to autism. There's lots of other reasons people can be quite suspicious of pharmaceutical companies that produce vaccines as well. So there's all of those. But the science and the examples in the past say that vaccines are really effective against diseases. What does antibiotic resistance mean? Oh, okay. Let's talk about that then. So let me find my slides on antibiotic resistance. Here we are. Okay, so here's our antibiotic resistance slide. This is what we talked about there. So, pathogens that are caused by, why would someone cheat results if they have no personal gain? That's an interesting question. Occasionally, scientists can feel pressure to show the results of the study that they think would be most popular. It's the way to get published. If you're publishing a novel, something that could be described as a novel result that would be really interesting to the public. So it can be easy to then misinterpret your results because you're fairly sure that they show this and it's very exciting, but actually that's not true, and you need to be really careful that you follow scientific procedure all the way through. Right, back to antibiotic resistance. So, pathogens that are bacterial pathogens and that cause diseases can be controlled by antibiotics usually. So they work very well. They tend to target the cell walls of bacteria that are causing disease. So they tend to be quite specific. You tend to have one antibiotic for each specific type of bacteria. Okay, so the bacteria grow and reproduce inside the body. A pathogen, they may cause disease. If you add the antibiotics kill the bacteria, and therefore you start to feel better. There's less of them in you. Keep taking the antibiotic to the end of the course, and after 1 week, they're usually 1 week antibiotics, all the bacteria have been killed and you've made a full recovery and you've left no survivors as such. If, however, bacteria grow and reproduce, you use an antibiotic, and by chance, one of those bacteria has a mutation that allows it to survive. It doesn't respond to the antibiotic, and so it carries on. You keep taking your course of antibiotic because you're good, take it the full course. But at this point, it's a bit late. The mutant bacteria is able to grow and reproduce in the presence of the antibiotic. The non-mutant bacteria have been killed because the antibiotic has still worked successfully on those. But because that specifically mutated bacteria could survive and reproduce, this is evolution, but fast because it's bacteria, they reproduce quickly, the population of the resistant strain rises. So you get more and more of the red bacteria that you can see here. This process is called natural selection and evolution. Antibiotic resistance is what happens when this happens repeatedly, and eventually the antibiotics are no longer effective. So that's what antibiotic resistance is. What can we do about it? Well, let me double check. What my slide said about that. Copy. Okay. So we can look at the causes of antibiotic resistance, which are overuse. So if patients go to the doctors and demand antibiotics for a viral disease such as a cold, the antibiotics are wasted and the bacteria have more chances to become resistant to it, to show that mutant strain. If you fail to complete a course, so often when you feel well again, after most of the bacteria in you have been killed, you might think, oh, I don't need these pills anymore, I feel fine, my immune system can do the rest. But that doesn't really work. So that leaves some bacteria alive that may mutate and may become resistant. So it's better to take the full course, make sure you've got all of the bacteria. And there's also, so in the past, we used to use antibiotics a lot in agriculture to keep animals healthy. It was an animal welfare concern. They would grow faster and you could then be sure that all of your animals would go along for, say, beef production if they're cows or milk. So this may lead to a spread of resistance from animal to human host. So all of those cows or livestock being vaccinated, you're more likely to see those strains of resistant bacteria because every animal is being vaccinated. So there's more and more chances for the bacteria to become resistant. And it may lead to a spread to humans. So now we only use antibiotics on animals that really need them. So if an animal is sick, then you would use an antibiotic on it. But if it's not, there's no point. So only take antibiotics when necessary, not for viruses. Use specific antibiotics, so make sure that you're using the ones that are specific to the pathogen that you need to defend against. Keep hygiene levels high, that just prevents bacteria in the first place. And isolate anyone who's got resistant bacteria. So quite often they'll do that in hospitals if they discover that someone's not responding to treatment. Okay. Good question there from Summer again. What happens if you have antibiotic resistance and what will the doctors do next if the antibiotics aren't working? So normally they'll try a different set of antibiotics. So you can sometimes approach bacteria from a slightly different angle, if you see what I mean. So a different set of antibiotics might target a different, say, part of that bacteria. So that's usually what they'll do, or they'll try other methods. Antibiotics aren't the last line, there's other things to do as well. Okay. Any more questions about the content, or should we go on and do some past paper questions and do some exam technique? That'd be fine as well. I'll just give you a couple of seconds. Feel free to put any questions that you have in the question and answer. We can always go over them if we need to. So someone's just asked, antibiotics can kill good bacteria as well. So what are the side effects of using them? They can, but antibiotics do tend to be quite specific. And so that means that they're more likely just to kill that bad bacteria. In contrast to when you are, again, working in agriculture, but say more on crops, so if we talked about plant diseases, if you use an insecticide or a fungicide, or any sort of herbicide on your crops, so that's generally what you use as a pesticide, and they might tackle different sorts of pests, they don't tend to discriminate between good and bad levels of the pathogen that they're targeting. Whereas antibiotics in general do. Not always, but in general. Another good question here. I think I know a lot about the topic, that's good, but I struggle to put my knowledge into an organised answer. What advice would you give? Oh, that's really tricky, that's really hard. So when you are approaching an exam question, I'm going to assume this is for the longer questions that we've talked about, so those 4 or 6 mark questions where you have to make your points in order. There's no reason why you shouldn't scribble a little bullet point plan below, where you've gone, oh, I know I need to talk about this, so I'll put it in a bullet point below. And then you can order them and then write them out as full sentences in the answer space. That's a really good way of tackling that. The other thing is making sure that you take time. It's really hard in an exam because you tend to feel quite rushed, but it's important that you take an extra 5 minutes or an extra even 10 seconds just to think through the answer and make sure that it makes a logical sense. The other thing is examiners are nice, generally, depending on how many papers they've marked that day. But if you've made a point and you've made your points well, if they're in slightly the wrong order, then you might be lucky and they'll give you the point. But the best thing to do is to have them in the right order and think them through. And practice. Do lots and lots of practice questions. And there's no reason for that to be a difficult thing. You can just do 2, and they don't even need to be a whole paper. You can find questions that are for specific topics. There are sites that you can go to, such as one I quite often recommend is Physics and Maths Tutor. They have questions by topic. So you can just do a couple on a topic that you want to revise, and the mark schemes are there as well. Get used to looking at mark schemes. It's really important that you understand what they might look like and what they're set out to look for. So on that note, let's have a look at some exam questions. So I've started with a fairly simple one here. So the MMR vaccine is used to protect against measles. Apart from measles, I'm going to get my pen out so we can underline keywords. So apart from measles, which 2 other diseases does the MMR vaccine protect against? Okay, good. One's popped mumps and rubella in the chat. That is exactly what the other 2 are. Okay. And then it moves on to talk about measles in the context of a vaccine. So measles is a dangerous disease caused by a vaccine, caused by a virus, not a vaccine. This is why you should read the question properly. Normally, MMR vaccinations are given at 1 year old and again at 4 years old. Each vaccination is 90% effective in protecting against the measles virus. In April 2013, there were 630 cases of measles in children aged 4 and over in a small area of the UK. These cases, 504 children had not been vaccinated against MMR at all, and only a few had been given a second vaccination. calculate the percentage of the children who caught measles in April 2013 who had not been vaccinated against MMR. Okay, so this is quite a lot of information in a short period of time, but we know we've got 630 cases. Of them, 404 had not been vaccinated. So we always do the smaller number divided by the bigger number. So 504 out of 630 cases, 504 divided by 630, and then times that answer by 100 to take us to the percentage. So that gives us the proportion, and then timesing by 100 gives us the percentage. It's 80%. Okay. See whether I've written the answers to this. I have. So suggest one advantage to the population. So we know we need one advantage to the population as a whole of children having the second MMR vaccine. And then what does the vaccine contain? So those are two things that we've talked about today. Okay. So here's the answer now, both 1 mark. So they're very simple questions, quick answers. So less chance of a pandemic or less chance of the spread of measles, mumps and rubella is a big advantage to the population of children having the second MMR vaccine. And someone's also said that having herd immunity, that would definitely be an advantage. And what does a vaccine contain? Dead or inactive pathogens, viruses, or bacteria, depending on what's needed. So yes, an altered or weakened version of the pathogen is another good way of describing that. Any of those would get you the mark. Okay. Here's a good 3-mark question to put into practice what we were talking about putting knowledge into an organised answer. We've got 3 marks here. Explain how a vaccination prevents infection. So we've used underlined explain there because that's our command word here, and explain means that you do need to give a more detailed answer. They want to know how a vaccination prevents infection. Be careful when you come to a question like this that you don't just blurt everything that you know about vaccination. You've already answered a question saying what is a vaccine, so you don't need to cover that bit. You need to explain how it prevents infection. So we're talking about how it affects our immune system. Because it's a 3-mark question, we know that we need to include 3 parts in our answer, or 3 parts to the explanation. So the way I usually approach these is to start at the beginning. So what happens when you're vaccinated? So when that weakened pathogen enters the body via the needle, the first thing that happens is that white blood cells produce antibodies. They do that in response to the antigens on that weakened form of the pathogen. So our white blood cells produce antibodies that are complementary in shape to those antigens. And so that makes them specific to that particular pathogen. This means that the response is rapid on reinfection. So we had that graph on the page a few minutes ago. So if you're infected again with the full form of the pathogen, you have a much steeper curve on the graph. So the response is much quicker, and there's also more of it. When antibodies are remade on reinfection, the antibodies help to kill the pathogen. And they do that by clumping the pathogen together, and then the phagocytes coming along and engulfing it. But we didn't need that huge level of detail that I've just talked about as part of that question. So I've simplified it down in this model answer. So the first mark was for white blood cells produce antibodies. The response is rapid on reinfection, that's another one. And then why is it rapid on reinfection? So the antibodies help to kill the pathogen. Another mark there. Okay, a 2-mark and a 1-mark question here, this time on antibiotics. So when I was preparing this topic, I thought, well, I reckon most of the questions will be about vaccination or antibiotics. So that's what I tailored the past paper questions to as well. So antibiotics can only be used to treat some infections. Explain why antibiotics cannot be used to treat measles. Okay, we've talked about this before. Keywords there are explain, cannot, and then measles. Measles is a virus. I've never been very good at writing with the crayon on my tutor. And part 2 is why do antibiotics become less useful at treating an infection if the antibiotic is overused? Keywords there, I suppose less useful at treating an infection, overused. So we talked about this today as well. Antibiotics don't kill viruses because the virus lives inside the host cells. So 2 marks. Antibiotics don't kill viruses was 1 mark, because the virus lives inside the host cells was the other. Bacteria may develop resistance to the antibiotic was the answer to part 2. Again, 1 mark there. Nice simple question. Okay, let's do 1 more and then we'll answer some questions because I can see them popping up out the corner of my eye. Plants need mineral ions for healthy growth. Which part of a plant takes in mineral ions? So we haven't really talked about plants today, actually. So this might be a good opportunity to discuss them if you want to. We did talk about this last time. Really good answers coming up there. I can see that you're saying that my voice is freezing occasionally. It should stop and if you let it carry on a little bit. If it does keep happening, then let us know. Roots, chlorophyll. Which part of a plant takes in mineral ions? The correct answer is the root. So we did do a session a few weeks ago when we were on the previous topic about plant organisation. So we talked about how minerals travel up and down the plant, how water travels up and down the plant. And the minerals start their journey at the roots. Leaves are usually green. What is the green substance in leaves? Good, chlorophyll. They could have been really mean here and put chloroplast, and then you'd have to have properly read it and make sure that you're circling the right word, not just a word that looks like the right word. Other words there are glucose and starch. So glucose and starch are both present in leaves. Glucose is the product of photosynthesis. It's also what we use for respiration and what the plant will use for respiration as well. And starch is a way of storing. It's a long chain of carbohydrates, is what starch is. That's how carbohydrates are normally stored. Plants are quite starchy. They have a lot of starch in them. Okay, let's have a question break before we do that next one there. Okay. Millie, I can see my voice is still freezing a bit for you. It's probably an internet problem. Is probably the best answer I can give for that. So see if you can move a bit closer to the router. Okay, let's do Couple more questions. We've got 15 minutes left. Green substance in leaves is important to plants. Explain why. Okay, that's quite a good one. So we're going to be going through this in more detail, probably in 2 weeks' time. We're going to talk more about photosynthesis and exactly how it works. But the green substance is important for photosynthesis. So the green substance is chlorophyll. It absorbs light for photosynthesis to make glucose for plant functions. Something that you'll probably be familiar with. Chances are, if you're not, and you're right at the beginning of your GCSE journey, then we will talk about that in more detail, so don't worry. Last one here, talking a little bit more about plant defenses, which we talked about on Monday this week. So, or was it Tuesday? It might have been Tuesday. A shortage of mineral ions can affect a plant. Draw one line from each mineral ion to the effect of its shortage. So we talked about two deficiencies that plants can have. We talked about deficiencies in nitrate and deficiencies in magnesium and how they affect the plant. So in both of them, we talked about how the plant kind of becomes a bit droopy, leaves don't work as well, photosynthesis doesn't work well, growth is impaired. But here they're asking you to split that out a bit. So they're testing quite a specific bit of knowledge here. Magnesium is most noticeable effect is stunted growth, whereas nitrate, the most noticeable effect is the yellow leaves. So nitrate goes straight for the chlorophyll. It stops the chlorophyll working, stops those cells being green because it's taken the chlorophyll out. And so the leaves go a bit yellow. They can't photosynthesize correctly, and that does affect the growth of the plant. Magnesium works in a slightly similar way, except it doesn't target the chlorophyll, but it still results in that stunted growth. So magnesium is essential for plant growth. They can't do it without it. Whereas nitrate, they can kind of cope for a while before it completely overcomes them. So magnesium is really essential for plants to pull from the soil for them to go anywhere, particularly for crops because they then feed us. 2 marks there. And that's all the ones I've popped in there. I can put some more in if that's what we want to do for the last 10 minutes or so. Good, loads of good answers on here, which is great. Any more questions about the content? Is there anything that you want me to go over again that you've thought, actually, I don't really know as much as I should about that? Pop it in the chat or the question and answer, whichever's easiest for this one. That's fine. Nothing there yet. Can I explain protists? That's a good one. Yeah, we can talk about protists. Any others while I'm loading up the slide? If you've got any other things that you'd like me to talk through, that's fine. Okay. So here we go. This is what the first slide where we talked about protists. Oh, good, Millie. I'm glad I'm not freezing anymore. So, a protist it's an animal. Well, it's a eukaryote, not technically an animal. So protists are very strange organisms that have features that belong to plants, animals, and fungi. They're not really any of them, but they have features common to them all. So they are eukaryotic, they have a nucleus, so they're not like bacteria. Bacteria are all prokaryotic. We're going to cover this next week, so if you're a bit confused about cell biology, then that's what we're going to do next week as well. So they're definitely eukaryotic. We can split them off from that part of the evolutionary tree. But otherwise, it's very difficult to classify them. And when I was preparing this lesson, it was very difficult for me to find a good diagram of them because they all look very different. Some of them have fungus-like features, so having a that type of cell wall that fungi have. Similarly, plants also have a cell wall. Some of them have ribosomes and mitochondria, others don't. They differ extremely, and they're all sorts of different shapes and types and structures. So it's very difficult to classify them. And that's why I think they're often quite difficult to remember because they don't have any particular features that characterize them. So they're this kind of funny group that fall in between everything else. And you don't need to know too much about them. You learn about them as part of when you're talking about classification and things like that. And when you're talking about disease, because one of the most popular diseases that's caused by a protist, and that comes up in every specification as far as I know, is malaria. Malaria is responsible for a huge number of deaths each year. It's very, I suppose contagious isn't the right word. It's easily passed on, and it's quite aggressive when it does strike. So it's caused by a protist called Plasmodium, and it infects humans, particularly in hot countries such as Africa or South America. The reason that it occurs in hot countries is because it's transmitted by mosquitoes acting as vectors. So vectors is when an organism carries a disease but isn't infected themselves. So mosquitoes are an example of a vector. Another example might be badgers and tuberculosis. So the mosquito bites an infected person, takes in a bit of their blood, gets a bit of blood on their proboscis, I think it's called, the nose or the mouth of a mosquito, and then they pass it on to the next person that they bite because they stick the same nose into the next person or the same mouth into the next person. How does it affect the host? Fever, sweats, chills, headache, vomiting, and diarrhea. So a combination of lots and lots of very uncomfortable symptoms. You can get dehydrated very quickly, and it really disrupts your immune system. There's no cure for malaria. You can only prevent it. So infection can be prevented by stopping mosquito bites. So that's why when you're in hot countries, you normally sleep under nets, and you often wear insect repellent, and you usually take anti-malarial drugs as well. So those drugs stop the symptoms and may also help to prevent the infection. So malaria, it's the symptoms that cause the trouble. So if you have consistent vomiting and diarrhea, you become dehydrated very quickly. Your cells can't cope, they can't get enough water to keep their structure, and eventually you may die. But not everyone dies from malaria. So it's very much dependent on each individual person. Plasmodium also counts as a parasite. That's interesting. I suppose it does, because it feeds on other organisms. Malaria is a deadly disease, and someone's immune system recovering from malaria. So the immune system can get rid of malaria if you've got it in sufficiently mild a version. And then your immune system, in theory, would remember it again. So there is some benefit to that. What is AIDS, as in a virus or a parasite, and HIV? So HIV and AIDS are linked. HIV stands for human immunosuppressant virus. It's a virus. It's caused by a virus. The trouble is, when you're infected by HIV, you don't tend to know about it. You might feel like you've got a cold, and then it'll go away, and you'll think, oh, I got rid of my cold, and that's fine. What it does in your body is it then very slowly attacks your immune system and suppresses it. It means that your immune system can't work as it normally would. Your white blood cells can't produce antibodies as they normally would, and other parts of the immune system that you may learn in more detail don't work. When it has got to the point where that part of that suppression of your immune system is starting to affect you, then it has become AIDS, which is autoimmune deficiency syndrome, I think that stands for. So HIV leads to AIDS. You can't have AIDS without HIV. But that first part, that infection, is caused by HIV virus and then develops into AIDS. And there's no cure for AIDS. You can just slow down the slow shutting down of the immune system. So that's how those two are linked. Good news is it's not as bleak as that. If a person has AIDS, we can slow it down an awful lot. So although there's no cure, they can still have a long life. Pop back to the slide about protists. Here. Okay, let's finish with another Slido, because I think that worked really well. So same question again. I'll just share it with you in the chat. So if you have a look in the chat, there'll be a link. There we go. So put that in. So I want to see whether this is any different now. So last time when we finished, we have lots of really good keywords. There was protist on there that got quite big, virus and bacteria. So what can you think of now? Can you think of any new ones to fill in that word puzzle? Is that working for everyone? Let me know if you can't see the link or anything. Good. Plasmodium, we didn't have that one before. What other ones have we talked about? Any plant keywords? So that link, if you copy that link and pop it in your browser, then that should take you to a question. You can put in a response as many times as you like, and it will fill in this word map here. It's a really good way of judging kind of what keywords are come up most in people's minds. Good. Deficiency, magnesium, spot, yes, rose black spot virus. We didn't really talk about that today, but that's one of the viruses that we talked about connected to plants rather than humans. Chlorophyll, we talked about a lot today, how that's key to a plant's growth. Mumps, yes, good. We hadn't really talked about that before. MMR vaccine, yes. Malaria, again, we talked about. Insects, good. All protists have a nucleus, yes, really good. They're all eukaryotes. Anyone put the eukaryote in there? That'd be great. Smallpox, good. That's another one we didn't really talk about today, but that's another one that's a good success story for antibiotics and viruses. Vaccination. Nitrate, good. That was the other deficiency that we talked about. Ash dieback, yes, we talked about that on Tuesday. That's something that I come across almost daily in my job, is talking to farmers about whether they've got ash dieback. What they need to do about it if they do. Eukaryotics in there, spelled correctly, brilliant. Mumps, another one there. Antibiotic resistance, vaccination, protists, really good. See, this is completely different from what we had at the beginning of the lesson, and I think that's really good. Waterborne, yes, good. I think that's good. So I think this lesson has brought back lots of the knowledge that we've talked about. I feel like we're quite ready for exams now, hopefully. Any more? Herd immunities in there, good, really good. this is completely different. Really nice to see. Okay, so I think we're going to have to stop there for that one. I'm going to stop it now. Return to here. So the next biology lesson will be next week on Monday and Tuesday at 3:15. It's going to be biology, and we're going to talk about cell biology and different types of transport in cells, so osmosis, active transport, and diffusion. We'll talk about next week as well. So join in those if you'd like to, and if you think they'll be useful. I'll try and make them as fun as possible. In the meantime, if you do feel like you need more one-to-one help, then my tutor offers specialised tutoring, so one-to-one tutoring with a tutor that you can pick. You can find tutors on the My Tutor website. Okay, anything else to add?
Not at all, Charlotte. You said it all amazingly well. Thank you very, very much for an amazing lesson. And thank you guys for joining us. Have a lovely weekend, everyone. Goodbye.
Bye, everyone.